one of the big surprises of the genetic and genomic era is just how much of our DNA is shared with all other animals, really. Right. It depends on how you do the math exactly, but something like half of the genes that the worm uses are genes that we use too, right? So we're something like half worm, if you like. Yeah. The basic building blocks are all there.
I'm John Foxe, Director of the Monte Institute for Neuroscience at the University of Rochester, and I'd like to welcome you to another episode of Neuroscience Perspectives. Today I'm absolutely thrilled to be joined by one of our own here at the University of Rochester, Dr. Doug Portman, who is the Donald M. Foster Professor of Biomedical Genetics, Biology and Neuroscience here at our medical center. He is also very recently elected a Fellow of the American Association for the Advancement of Science, and his lab uses C. elegans, microscopic roundworms, to study our understanding of the genetic underpinnings of sex differences in neural development, behavior, and disease susceptibility. He's also a renowned trainer of trainees here at Rochester, providing guidance and inspiration to our fellows and to our students, and we'll get into all of that with him. Doug, thank you so much for being here.
Oh, thanks, John. It's a privilege and an honor, and I appreciate the invitation.
Well, listen, I was thinking about this, you show up at an event and somebody says, what do you do? I'm a geneticist, I'm a neuroscientist. And then the next piece is going to land on, I work in worms. And I'm sure every now and again you get one of those looks like, what?
What? Yeah.
So why worms? Hit us with the...
Yeah, good question. Yeah, that's a good place to start. C. elegans is an amazing model system that has provided numerous profound insights into lots of different areas of modern biology. There are a number of reasons why it's a very powerful system. We can talk about some of those in detail if you'd like to. I can talk about worms all day. But I guess the quick answer to that is that worms allow us to approach important fundamental biological problems at a level where we can sort of view them from, I don't want to say necessarily a reductionist perspective, but from a reduced perspective. Right. As somebody who trained originally as a biochemist, my mind thinks, sort of bottom up. And C. elegans in many ways is sort of the simplest instance of a creature that has extraordinary, fascinating, beautiful biological complexity. And so that, for me, that's the reason why why we study it.
So it's, in some ways, it's stripping away all the noise of complex, super complex creatures like ourselves, our first cousins in the primate. And I see our backdrop here has some of these. Most people, they show the picture of their puppy or their cat, but these are your pets right here.
Absolutely. Absolutely. I feel very close to these creatures.
But it's that simplicity of the organism, and yet the behaviors are profoundly complex, right?
Yeah. In fact, I tend to shy away from the word simple because, C. elegans is a simpler organism. It's a reduced organism, but it's still extraordinarily complex. And there are lots and lots of things that we are just in some ways fundamentally clueless about in terms of the way it works. So yes, behavior, the function of the nervous system, developmental programs, lots of aspects of biology are extraordinarily complex, even in this sort of highly reduced context. And for me, that makes it a great place to sort of start. If we have any hope of understanding at a really high resolution level how the human brain works, how it integrates information, and where our consciousness comes from. If we can't understand how neural circuits work when we're talking about a few hundred neurons, then there's a fundamental disconnect, and we still have some work to do.
Yeah. And just for the viewer out there, they say, well, what the hell is a worm to do with a human? But we have a lot of shared biology, right? And a lot of shared genomics or genetics.
That's right. In fact, one of the big surprises of the genetic and genomic era is just how much of our DNA is shared with all other animals, really. Right. It depends on how you do the math exactly, but something like half of the genes that the worm uses are genes that we use too, right?
Yeah.
So we're something like half worm, if you like.
Yeah.
The basic building blocks are all there.
They're all there,
and maybe they work in somewhat simplified ways, but yeah, the same genes that specify the difference between our head and our tail, our feet, during development. Work in a worm. The same genes are involved in, specifying, telling cells to become different from each other during development. The very same genes.
Right. And I know, in some ways, I think this is a concept that makes a lot of people a little bit uncomfortable, right? I mean, and obviously there's been lots of controversy, but, that we're all part of the same biological chemistry. And
that's right.
And again, going back to that business of this tiny little creature that you can literally see through, and just how easy it is then, or not easy, nothing's easy in our world, but how much easier it is to actually get a tractable question about a specific gene and that.
That's right.
It brings me to two thoughts, actually. One is you mentioned a few hundred neurons, and I happen to know you trained with a very eminent worm biologist, worm neuroscientist, Scott Emmons,
Indeed.
who we both know very well.
Yes.
Scott's really famous for literally mapping the entire neuronal network of the C. elegans. Can you tell us a little bit about that? Tell us about your time with Scott and how that played into your career.
Yeah, well, I can say a bit about training with Scott, which was a wonderful experience, and then I can also say a little bit about this, the value of the mapping that Scott and others have done, and how it's sort of opened exciting new doors for the field. So, I decided to train with Scott because I came from a background. As I mentioned before, I sort of trained as a biochemist and cell biologist.
Yeah.
And
at the University of Pennsylvania.
That's right.
That's where you
that's
did
right.
For undergrad and your grad. Yeah.
Exactly right. For both undergrad and graduate school. And I loved my time there. I learned a tremendous amount. I felt like I was able to do some good work. But I got to a point where all of the experiments that I were doing were mixing things together in test tubes and running gels and sort of, doing things that were very, in some ways, divorced from real biology. And I wanted to get my hands on a living creature, but a living creature in which we could really, we really had the power to understand at this very basic mechanistic level how genes and molecules work. What is the function of those individual components that we can study in isolation, but how do they work together to build an organism that develops? So I decided that C. elegans was an ideal creature in which to do that. And for personal reasons, I was interested in staying in the New York City area in the Northeast. And Scott became the sort of immediately most logical candidate to train with. Scott has been in the worm fields really since its founding. And there aren't very many people really who can say that anymore. Scott has a tremendous perspective on the holistic perspective on the field and understanding where it started, how it got to be where it is, who's contributed what. And that was really valuable for me in my training because it's given me a really solid foundation in terms of just kind of understanding the system without necessarily diving immediately into some little detail, but to be able to understand it at a holistic level. And so Scott was really instrumental in kind of instilling that kind of a view, I think, in me and all of us who worked with him.
Give us a number. So I work mainly in humans, and I'm stuck dealing with 70 to 100 billion neurons in this extraordinary connective mass.
Indeed. Yes.
And so of course there's an intractability to the complexity of the circuits that make up the human brain. I shouldn't say intractability, but they're difficult to study.
Sure. But
the number is in C. elegans?
Well, the number depends on which sex we're talking about. In C. elegans, there are two sexes. They're not male and female. They're male and hermaphrodite. Yeah. So the C. elegans, essentially the female has learned this little parlor trick over evolution of being able to make its own self-sperm. So it's a self-fertile female, if you like, but we call it a hermaphrodite. Yeah. For many years, we've known that the adult hermaphrodite, has exactly 302 neurons.
302 neurons.
The male has somewhat more than that. The number is about 395. You alluded to the connectome before. One of the initial visions for C. elegans when it was first being established as a model system in the late 60s and early 70s by the great late Sidney Brenner was that he could imagine that in a system like this, that you might have the opportunity to really get a level of anatomical description of the nervous system that's unparalleled in any other system, to identify every single cell. And not only that, to understand how they connect to each other, right? A neuron in isolation doesn't do very much, right? The brain works, as you alluded to, because neurons are connected into circuits that can do all kinds of amazing things. And so actually there was this initial feeling that once if the field were able to do this thing, which at the time was a tremendously daunting task of identifying all those cells and identifying all the synaptic connections between them, that the answer would fall out, that we would understand how it works, that the structure would lead immediately to
an aha moment.
Exactly, to this eureka, I see, I get it, now it all makes sense. And unfortunately, something like the opposite happened. So the hermaphrodite, we call this the connectome, right? The wiring diagram of the entire nervous system. The connectome of the adult hermaphrodite was an arduous task led by John White at the MRC, done mostly in the mid-'80s. So we've had that information for quite a while.
Right.
And I think one of the really astounding things about that is that it's a tremendously valuable tool, but in itself, the structure of the nervous system is not sufficient to tell you about its function. And so that has been, the field has been struggling with that ever since.
Maybe a good thing in terms of like if this eureka moment came, we'd all be out of business.
That could be true. Right. And so an important advance in that field has been, you mentioned Scott Emmons' role in this. Scott was the first to really decide to tackle the nervous system of the adult male. And after the adult hermaphrodite had been finished in the 1980s, it was a tremendous task, and they sort of breathed a big sigh and said, "We're not doing that again."
We're done, yeah.
Yeah. And it wasn't until there were some technological developments, and also Scott just had the patience and the fortitude and the drive to get it done. And over the past 10 years, has spearheaded the effort to drive the connectome of the adult male. And now, because of additional technological advances, we have multiple connectomes. We have connectomes for multiple developmental states, multiple individuals of both sexes. But we're still stuck at this place where having the connectome itself is a really valuable foundation, but it doesn't in itself tell us how behavior works.
But it gives you that ability then, right? And we should mention that all of this work was done in the Bronx, in New York, right? At Albert Einstein College of Medicine.
Indeed.
Placed near and dear to both of our hearts.
Yes.
But having that wiring diagram so precisely mapped then allows you to focus in on a neuron or a cluster of neurons and really ask very specific mechanistic questions about what this piece of the circuit or that piece of the circuits does, and that has a level of tractability in a creature with 390 neurons that it's just utterly impossible in almost every other species, right?
Yeah, that's right. I mean, I think of that. In a sense, you can divide that up into two overlapping halves. One is, how does information in the genome specify the identities of all those neurons and specify their ability to wire up into circuits? And then how do those circuits do what they do, right?
Yeah.
How do they take in and integrate and process information and choose to make a decision about executing a behavior, and then how does that behavior actually get executed? Right. And we can approach those questions at the level of single neurons, single synapses, single genes, in a way that has not been possible really in any other system.
Right. It's absolutely extraordinary. Going to some of the specifics of your work, like you're very interested in this issue of sex differentiation, and and you have these two sexes, a male and a hermaphrodite. And the programming of these genes can really lead to very different types of behavior in terms of like feeding versus mating and so on. Would you tell us a little bit about that and what you've learned and the kinds of systems that those urges or desires rely on?
Sure. So, we've been interested in trying to use biological sex as an entry point to understand sort of where there are points of plasticity in the development and function of the nervous system that can lead to adaptive differences in behavior. And so, because of this extraordinarily high level of resolution with which we understand the structure of this elegans nervous system, we know exactly what those sex differences look like. We know that each sex has its own set of sex-specific neurons and sex-specific circuits that are dedicated to purely sex-specific behaviors. Hermaphrodites have to lay eggs. Males don't. Males don't have eggs, right? And so there's a dedicated circuit in hermaphrodites that controls egg-laying behavior.
Right.
There's a dedicated circuit in males that controls courtship or mating behavior, which is a behavior that hermaphrodites don't do.
Right.
What my lab has been interested in is a discovery that came out of a serendipitous observation, actually, when I was first starting the lab here in Rochester, that led us to wonder about what about the rest of the nervous system. 294, exactly 294 neurons are shared between both sexes, right?
Right.
And that means they're lineally, anatomically, morphologically, essentially equivalent, right? So most of the worm's brain is the same as it is in most species. And yet what we found is that those shared circuits can be tuned in different ways to allow animals to prioritize differently among shared behaviors, or to execute those shared behaviors in different ways, such that the features of those behaviors lead to differential outcomes. So an example of this is that often hermaphrodites will tend to prioritize feeding behavior, because for a hermaphrodite, fertilizing your own oocytes and producing progeny is an important part of your life. For a male, males need to find mates, and so males will often sacrifice their ability to feed and go out and search to search for hermaphrodites, often abandoning a food source to do so. And so those aren't sex-specific behaviors necessarily. They're just really differential modulation of feeding and exploration. Both sexes can do that. And so we're interested in kind of finding the knobs and dials that evolution has engineered that permit the sexes to do those different kinds of things.
So that the male biases towards looking for a mate at the expense of feeding.
That's right.
And essentially the risk ratio there is quite different, right?
Indeed.
And the risk, it's maybe a proxy for risk taking in a worm.
In fact, we do sometimes think of it that way, that males are a bit less risk averse than hermaphrodites are. Right. And so some of the things that we've been doing in the lab recently is trying to ask questions like, how willing is a male going to be to cross an aversive barrier of something that tastes terrible or is toxic compared to a hermaphrodite? And does that differ depending on whether an animal is hungry or not, or whether it's mated recently or not? And if those, we have some instances where it seems like there are some interesting things going on there, and then we can go in and ask, where in the nervous system are things being modulated to bring about those outcomes?
Amazing, right? A few hundred neurons, 80 billion neurons. Very, very similar behavior in males.
Absolutely. Yes.
It's extraordinary. And remind me now, maybe I'm wrong about this, but actually quite a lot of this relies on the chemosensory system, right? The smell system, and that you see these changes in genes coding for smell that actually change risk-taking behaviors.
Yeah,
if I could put it that way.
That's right. And that, I think, is one of the most interesting and unexpected things that we've found in the lab. When we started this work, I guess my intuition about scientific problems is often completely wrong, and this is a good example of that. We imagined that the way that animals were going to be prioritizing different behaviors differently according to their internal and external needs and signals, the way that would work, at least naively to me, I would imagine that evolution would design an animal so that it can gather as much information as possible about its world. And then, fancy things happen in neural circuits, inputs get weighed, and essentially a decision gets made about what to do. And so according to that, we expected to find that the sex differences would be in neurons that are a few layers deep in the system, in interneurons that are integrating information. And we might find that, maybe one input is a little bit stronger in a male, or a particular neuron is modulated differently to bring about a different behavioral outcome. And I think that probably is happening, but we haven't really found any good examples of that. Instead, what we've found is that the way evolution has engineered in the ability for animals to behave differently is by front-loading it at the very sort of the entry point to the nervous system, the sensory system, and in particular, the chemosensory system. Chemosensory behavior, smell and taste, are essential to a worm. They can't see, they can feel a little bit, but really their primary mode of gathering information about their world is smell and taste, right? Does this taste like something I should eat? Should I run away from this? Is it toxic? Is it a predator? Is it a potential mate? And so there are chemical signatures for all of those things.
Right.
And the worm sensory system has specialized sensory neurons and receptors, individual proteins within those neurons that detect all of those stimuli. And what we found is that the expression of those receptors that detect those stimuli can themselves be changed by biological sex and also by things like feeding status.
Right.
So for example, an adult male, one reason that it's able to leave food is simply because a receptor that detects the presence of food is downregulated in the male. So males just don't smell food as well as hermaphrodites do, but they smell sex pheromone much better than hermaphrodites do, right? And so rather than gathering all the information that you can and making a decision, evolution has sort of forces the worm to pay attention to what it has decided is the most salient cue for it according to that time and stage in its life, which I find fascinating.
Absolutely astounding. It reminds me actually of, you're talking about evolution as this force, which of course it is over time, but a lot of it was Stephen Jay Gould who used to talk about not adaptive but exaptive functions. This idea that random stuff happens in the genome, like downregulation of a chemosensory receptor, and then those few worms that have that downregulation are more likely to drift away from food, and they're going to be more successful in identifying mates, and it's just happenstance. But then of course becomes reified in the genome. I mean, it's extraordinary.
Absolutely.
Really beautiful examples of that. We'll switch gears a little bit. We'll stop geeking out about the worm just for a moment. You and I have known each other best part of a decade now, and I know some of our earliest conversations were around students and trainees, because at the time you were really helping direct our graduate program. Right. And I think I have a couple of questions for you. Like you are an extremely dedicated mentor and very involved in the student body. I'd like to know about your motivations there. And then I'd also like to ask, as an addendum, have you seen change? And is that change for the better over the last decade or so in the makeup of our classes, in the way in which we're approaching the academic efforts, the recruitment? That second question is a tricky one. The first one is a bit easier for me. My motivation, right? I find interacting with trainees to be one of the, if not the most rewarding part of doing academic science, right? We get to, interact with bright, young, energetic, creative people all the time. And for me, that's it's a tonic, right? Yeah. It keeps me from getting old. Yeah. And so I find it really gratifying to see a student come in, like many students coming into a PhD program, maybe somewhat unsure of themselves or what they're interested in, not sure if they're really up for this thing that they've signed up for, and then thrive and really just blossom and go on to do fantastic things. And so that playing some small role in that is an honor and a privilege. Right. So that really is what drives my interest in mentoring. I think you raise a really interesting point that I think graduate training has changed, certainly since you and I did it. Mm-hmm. But even over the last 10 years, I mean, I think there are a number of reasons for that. One that I think about a lot is that the amount of things that a student has to learn in order to master a field has grown exponentially. Oh, yeah. Right? When we... I don't know about you. I remember being in grad school and being overwhelmed by the literature. Yeah. Right. How can I possibly master all this? Small fraction of what it is.
Tiny fraction. Right. Yeah. It's a tiny fraction. And so now students will come in and I don't even know where to start sometimes with, in terms of getting them up to speed, not only on our field, but on fields that, is essential as a scientist to know about other things so that you can find those connections.
Intersectionality now. In the old days, you could learn microscopy around this system, and that's what you did for most of your career. There's no way to be that specialized today. I think you can look at this in different ways. And sometimes I get concerned that students become hyper-specialized too quickly because the frontier of
by its nature, as science advances, we drill down deeper and deeper into these sort of veins of gold that we found until you find yourself sort of at the bottom and you can hardly see up again. And I think it's really easy to fall into that and not be able to step back out and understand why did you go down that vein in the first place? What was the point of studying that, and how is it connected to all of these other things? And I think, exciting advances in science so often come from interdisciplinary kinds of thinking. People, some of us are lucky enough to have had the opportunity to train in multiple fields or have interest in multiple fields and be able to see those connections, but also just to be able to interact with people regularly whose perspectives and whose background are a bit different than yours, I think is tremendously stimulating. And we need to foster that in the next generation of scientists. And I get concerned sometimes that we focus so much on, you need to learn your field, you need to focus on doing your experiments, that we don't give students enough time to step back and think about the bigger picture and understand why they're studying what they're studying. They're not studying, we all study our own individual little problems, but ultimately we shouldn't be studying them for their own sake. We should be studying them because they're a critical piece in this larger puzzle. And if you don't understand, if you can't see the larger puzzle, you're lost.
Right. That's very well put, yeah. Talk to me, so if you don't mind me getting a little personal, we've had a huge drive here at Rochester, but I know most of the great institutions in the country have really worked hard on diversifying the student body, diversifying the faculty body, and that. And you're from a minoritized community in the LGBTQ world, and that. How's that played into your career? What have been the challenges? What would you say to a young LGBTQ person coming into the field today? What are the minefields? What are the advantages?
Yeah, great question. I feel really privileged and fortunate to have always had training experiences. In my training, I was always in very supportive environments. I've been out and had a partner since I was in college. And, so being out in grad school in the early 90s that was a different time.
Yeah.
And in fact, it was unusual to encounter another person from the LGBTQ community in science, or certainly a person who was, sort of out and open about their situation.
Understood. Yeah.
It's tremendously gratifying to have seen that change so much, right? We have an abundance of trainees in our programs here in Rochester who make up this beautiful, diverse spectrum of people in the LGBTQ plus communities. And but I guess I would say that still we do know that LGBTQ people tend to shy away from careers in STEM a bit more than their counterparts, and they also tend to sadly drop out of their training more commonly than people from
what's
from
driving
other groups.
that? What's the root cause there? Is that
I don't know the answer to that. I think that our job I think the answer is going to be complicated, and probably there are a lot of case-by-case issues, but I think ultimately what we have to do is realize that our job as educators, right, is to help everyone reach their potential and be the best scientist they can. And that means welcoming and supporting and including all kinds of people. And even if we don't understand necessarily what we're going through, what they're going through, simply to provide that message that we're there and we understand that they may be facing special challenges, and we want to help them, and that we would hate to see somebody leave science for a reason that is ultimately maybe not the best choice for that person's future.
When going back to in the 1990s, and you're starting out on your scientific career, could you look to people in the field? I mean, I just think about this walking in the shoes, right? Could you look and say, oh, there's a successful person like me, or that was. Yeah, right. So
no, I remember.
Is that a key component here?
Well, so let me say, there's a big qualification to my no, because that did change at a certain stage in my career. But I think back to when I was an undergrad, and I was very involved in the LGBTQ student group at the time in college. but it was a different world, right? People's views on gay people and the role, the place of gay people in society was so different. Right? We had Anita Bryant out there, preaching homophobic messages that were resonating with, and lots of other things like that. I remember having. It
hasn't stopped.
No, certainly hasn't stopped. And for a number of unfortunate reasons, many of those voices have become amplified recently.
Right.
But I remember very clearly being in college, being a senior in college in my biochemistry senior seminar. And the professor, we were talking about fruit fly genetics, and there's an interesting fruit fly mutant where the males don't court. And it's called fruitless. And I remember the professor making a homophobic joke about the sexual behavior of fruit flies, and everybody in the class laughs, and nobody at that time would have thought about raising a ruckus or like that was just like, oh, ha ha ha.
Right.
So that, but
and that hits home though when you're sitting there as a gay person,
right?
Of course,
it says to me, like, is this do I belong here?
Right.
It really makes you sort of question what your role is, what your potential is.
Yeah.
So I mentioned there was a big asterisk. So, my graduate school training was great. There was a fellow person from the LGBTQ community in the lab at the time. It was a big lab, and so there was that nice support, and there was acceptance from friends and colleagues. When I went to do my postdoc, by happenstance, it turns out that my postdoc advisor is also a gay man, and so that was
and that was pure happenstance.
It absolutely was, yes. Yeah. And so having that experience sort of really cemented in my mind the idea that one could be a successful, and in the case of Scott Emmons, fantastic successful scientist, and still be a happy, healthy, productive gay man in society.
And a great runner as well, Scott.
He is.
That's really fantastic. And we're mentioning walking in the shoes, and I think what you just did, I know from folks in my own family who are gay, it's not easy because they grew up at the same time, and you have that ingrained in you, that sort of sense of threat and worry about being out and being open. But by being here with us and being willing to talk about that, you're also part of the solution. And I know I've been able to witness that as well with the way you deal with students and trainees and the community here in neuroscience at Rochester.
Thanks, I appreciate that. I mean, I think it's very important for those of us who are in a position where we can have some influence to talk about those things and set an example.
Well, Doug Portman, you do that in spades, and thank you for being here, and thank you for taking the time out with us. Really appreciate it.
Thanks, John. It's been a pleasure.